Welcome again. So this is the second part of my presentation about gene expression and its regulation, and this would focus on few applications and methods that are being used for the study of gene expression in a particular cellular situation. This mainly relies on data obtained by the ENCODE project, and I would like to briefly introduce the ENCODE project here. So these were Ewan Burney and Magdalena Skipper. Ewan Burney from the European Bioinformatics Institute. Magdalena Skipper is senior editor of Nature, speaking about the tasks and the achievements of ENCODE. And actually, ENCODE has identified major resources about the genomic organization and the structure of regulatory elements on the human genome, and has put the emphasis on the non-coding regions of the human genome. So just to briefly summarize the activities of the ENCODE consortium. So these are elements that they are searching for and that they have already identified, like these long-range regulatory elements, the cis regulators, which are involved, which are important for expression of genes, of course, the core promoter elements, and finally, structures within the genes, regulatory structures within the genes. And they are using a number of different methods and resources in order to identify these elements, and these methods are described here in more detail. These resources are then further assigned to functional units, and they are depicted in genome browsers. So in the UCSC browser in particular, these elements can be identified. So they are using their raw data, and their raw data mainly based on high throughput sequencing, and I will come back to that in a minute. Then they have the ground level of data where the different approaches identify various elements that are important for gene expression, and they have the integrative level where this information is then combined into the information about, for instance, cis regulatory elements and CTCF sites and other elements that are important for the expression of each single gene. So if we have an experiment, if we have a particular cellular situation, if you have a treatment, for instance, we would like to identify a particular expression profile. How would we do this? So of course, we have to keep in mind that we are dealing with changing expression levels. So we are dealing with changing amounts of RNA. The basic situation would be this steady state situation where we have a particular synthesis of RNA and a particular degradation rate of the RNA. And so in the steady state, we have a synthesis rate which equals the degradation rate. If we have changes, depending on changes in the cellular environment, depending on treatment, for instance, we might either have an increase in the RNA amount, so we would have an increase in the synthesis, or we would have a decrease, so we would have an increased degradation rate, which leads either to a new steady state situation or which leads to maybe a complete removal of the RNA from the transcriptome. So in order to identify these situations, these cellular responses, we have to keep a number of aspects in mind. And this is, of course, the importance of cell types. RNA is specific for a particular cell type, so we would never cover the whole transcriptome if we analyze a particular cell type. We have to keep in mind the state of the cell in a developmental situation in a particular environment. And we have, of course, the condition or the treatment of these cells to keep in mind if we analyze the RNA. So if we want to do a particular experiment in a particular setting or in a treatment, then we would have such a design of the experiment. We would have, for instance, two different tissues or two different types of cells in two different situations, and we want to compare these different tissues. And there are two major methods for this purpose. And this is either a microarray analysis of the transcriptome, or this could be a high-throughput sequencing approach with RNA-seq. These data would then be analyzed, and of course this has to be computer-based. We need high-performance computers for the analysis, which then leads to expression profiles in other data that would be also included into databases. So just very briefly about these methods, we could use the microarray analysis, and this is a good approach for the comparison of two different situations, of two different transcriptomes in one single experiment. So a microarray is hybridized in this case to two different cDNA preparations, and we would then scan at different wavelengths. We would scan this microarray and find either an increase of expression or a decrease of expression between these two transcriptomes. The other approach, and this is actually the most frequently used approach nowadays, is RNA-seq, so a high throughput sequencing based approach. With RNA, from the starting RNA, we would fragment the RNA. It is then transcribed into cDNA fragments, into double-stranded cDNA, and these double-stranded cDNA elements are combined with adapters and then sequenced in parallel. So we would have extracted poly-A RNA, for instance, if we're looking into protein coding genes, that is analyzed in this approach. So it is also important to note in this context that the RNA is analyzed indirectly here, because we always need the reverse transcription into cDNA in this approach. But this is still the most straightforward approach for the analysis of transcriptome changes or transcriptome stages in particular cells. Just to give one brief example, this would be the analysis, how we would find gene expression profiles. Here we have for one particular genes, for instance, the expression levels at particular time points, and we would typically have either a decrease or an increase of the gene expression, which is shown here by different colors. So we would, for the red color, means that we have an increase, a fold increase in the expression of this gene. And these expression values can also indicate the relatedness of different expression profiles. So we would find relatedness of different genes. This can be shown in this hierarchical clustering for the analysis. This is another example. This is an experiment about the cell type specific gene expression in scleroderma skin biopsies. And this is how the overview of this expression analysis would look like. This is an overview of more than 1,000 different genes that change more than twofold in samples from scleroderma patients. And in more detail, we would have such a picture here. They have tried to analyze these data based on particular cell types and for comparison between scleroderma samples and control samples. And this is just a magnification of this part of the overview here. These are the genes that identify fibroblasts. So this would be the fibroblast gene expression cluster. We, of course, see clear differences between fibroblast lines and identified by gene expression and other cell lines. And we have some differences between scleroderma samples and control samples. However, they are having the problem here that they can only indirectly identify particular cell types based on the expression patterns of these genes. So of course, the goal would be to more precisely identify particular cell types. And this is now possible with single cell analysis. And there are a number of approaches into single cell analysis. One of these is the RNA drop-seq. So the typical situation is we have a complex tissue like this, and we want to analyze specific cell types within these samples. So we would have to isolate single cell types in this analysis. We have these cell types here extracted, and each of the cell types would then be collected in a droplet. So we have a cell suspension with these droplets, and each droplet contains a particular cell and contains microparticles. And the RNA-seq, the analysis of the transcripts of this particular cell, is done on the microparticles within these droplets. And this would give us RNA-seq as an RNA-seq library with 10,000 single cell transcriptomes. So we would be able to analyze all different cells, specifically analyze all different cells which are contained in this complex tissue. Of course, this involves further steps, in particular a suitable barcoding system to identify these individual cells that are being analyzed in the analysis. But this is promising. This is still difficult, but it's promising to really analyze single cells for the transcriptome. So in the end, I would just present one story, one publication that uses the data that were identified by ENCODE, that demonstrates that these control elements, these regulatory elements outside the coding region and outside the co-promoter are important for diseases, also for single gene diseases. These are cases of the so-called keratolytic winter erythema, an autosomal dominant peeling skin disease, which is particularly frequent in South Africa. Cathepsin B was long known as a candidate gene for the disease, initially based on linkage analysis, but mutations were never found within this gene. So further analysis involved a genomic sequencing approach, and actually they found an overlapping region between the candidate region in South African patients and Norwegian patients, and this overlapping region is outside the cathepsin B gene. So we have the cathepsin B gene here, and this is the picture on the genome browser, on the UCSC genome browser, and we have the transcript here, the CTSB transcript here, and this is the overlapping region outside the coding region. I then looked into further characteristics of this region, and you can see in this overlapping region, there is a signature of acetylation at lysine 27 in histone 3. And this signature is characteristic of enhancer activity. So the duplication that was actually found, the tandem duplication that was found in this region in the genomic analysis involved an enhancer in this element. The signature of the methylation at K4, where we have a peak here, this identifies an active promoter, and this is actually then the promoter of the cathepsin B gene. So we have a duplication in an active enhancer region. Does this really have an effect on cathepsin B? Well, so then next they had to look into the topological domains, into the functional units, and these are identified by the CTCF sites. And actually there are 3 CTCF sites here. One is there, one is here, and one is here. And this means that the promoter of cathepsin B and the enhancer that is duplicated in these patients are in the same functional unit. So this points to an activity of this enhancer towards the promoter for the cathepsin B gene. In order to demonstrate that this is really the case, we would also need a functional analysis and activity analysis. And this is summarized on this final slide here. So we have the overlapping region here between the 2 groups of patients. We have the high level of K27 acetylation, which is characteristic of the enhancer. And they've also found polymerase 3 or mapped RNA polymerase 2 activity, and the RNA polymerase 2 activity is found in this region, which is the region of the cathepsin B gene. So is there a correlation between the enhanced activity and the transcription of the nearby genes? And this is actually the case. So we have the enhancer activity here. Enhancer is active, and it is actually active at the same time point during keratinocyte differentiation as the activity of cathepsin B. And this is what we would expect in such a phenotype. So the enhanced activity correlates with the activity of the expression of the cathepsin B gene. And this is not true for two other genes which are located close to this same enhancer region. So we have a non-coding enhancer which was identified in the ENCODE project, which is duplicated and which leads to overactivity, overexpression of the cathepsin B gene underlying this monogenic disease. And this nicely demonstrates that the identification of these regulatory elements and the identification of mutations or structural variation outside the coding region can be important also for monogenic rare diseases.